Intestinal coccidia are most prevalent in patients with AIDS or those who are otherwise immunocompromised. The diagnostic stage involves oocysts passed in feces, and the infective stage is also the oocyst. For Cryptosporidium parvum, infection occurs when infective oocysts are transmitted through the fecal-oral route. Remarkably, they are immediately infective once passed. In healthy individuals, the illness is usually self-limiting. However, in immunodeficient persons, the infection leads to severe diarrhea, fever, nausea, abdominal pain, and weight loss. Diagnosis is made with a modified acid-fast stain, where oocysts appear red against a blue background, typically measuring 4 to 6 micrometers. For Cyclospora cayetanensis, infection occurs when oocysts are ingested in contaminated food or water. Outbreaks often trace back to produce such as berries or lettuce, especially during the spring and summer months. The clinical disease is essentially indistinguishable from cryptosporidiosis. On modified acid-fast staining, oocysts range from light pink to deep red, with an average size of 8 to 10 micrometers, larger than Cryptosporidium. These organisms also autofluoresce under ultraviolet light. Cystoisospora belli, formerly known as Isospora belli, is transmitted via fecally contaminated food or water. Many infections are asymptomatic or self-limiting, though some may cause gastrointestinal distress or severe dysentery. Diagnosis is made using a modified acid-fast stain, where sporoblasts or sporocysts appear deep red. Finally, Microsporidia are obligate intracellular organisms, with at least 15 species recognized as human pathogens. The most common species causing enteritis in AIDS patients is Enterocytozoon bionucii. These organisms are very small, ranging from 1.5 to 4 micrometers. They have a unique feature: spores containing a polar tubule, which allows them to inject infective material directly into host cells. Infection is most likely acquired by ingestion of spores, though inhalation, ocular exposure, and even sexual transmission are possible. Clinically, the disease resembles cryptosporidiosis, and the spores are highly resistant. Diagnosis can be made with a modified trichrome stain, in which the spore wall stains bright pink while the background appears green or blue, depending on the counterstain. Serological testing and PCR are available in research settings, and electron microscopy is necessary for speciation, though not routinely available in clinical labs. When studying parasitology, it is important to remember that parasites are not only detected in stool specimens. They can also be found in a variety of other body sites. In the blood, we can identify Plasmodium and Babesia within red cells, Leishmania inside macrophages, and Trypanosoma circulating in plasma. In sputum, Pneumocystis girovetsii may be observed. In muscle tissue, particularly cardiac muscle, Trypanosoma cruzi may appear in its amastigote form. In the liver and spleen, organisms such as Entamoeba histolytica and Leishmania donovani may be found. On the skin, Leishmania species can be detected. In cerebrospinal fluid, Naegleria fowleri is a notable pathogen. In the eye, Acanthamoeba infections may be present. Finally, in urine samples, Trichomonas vaginalis may be recovered. The key message here is, beyond stools, parasites can be found in many other clinical specimens. Always consider the site of infection when selecting diagnostic tests.